بسم الله الرحمن الرحيم. السلام عليكم جميعا وأنتم مساء. I would like to introduce myself first. I'm Dr. Walid Al-Amam, the head of automated core facility and point of care testing at King Faisal Specialist Hospital and Research Center. I'd like to, on behalf of my colleague in Saudi Society of Clinical Chemistry, to welcome all the audience to attend this event with the cooperation with the Saudi Point of Care Testing Association and also cooperation with sponsoring with the ROCHE Diagnostic company. This night, the event about the third series with point of care, and the main title for our topic is coagulation. As we know, all of us, we have a good experience with the point of care testing in the health practice, the practice with the glucose and glucometer and urine diagnostic. But I think the interesting topic to talk about this night and this event about coagulation and how to implement the point of care testing in coagulation field, and what is the advantage and also the challenge in this one. Today, this night, I would like to welcome our speaker and expert. We'll start with Dr. Tariq Aweda from King Faisal, and then after that, lecture with Dr. Malak Al-Mish'ali from the military hospital of Prince Sultan Medical City. And at the last talk with Mr. Rani Bisiso from ROCHE Diagnostic company. So today, this night, we'll start now with the first lecture with Dr. Tariq Aweda. He will talk about the coagulation and point of care testing overview and update. Before we start with Dr. Aweda, we will just to make a quick introduction. If you know Dr. Aweda, I think it will take a long time to introduce him. He's very famous in the field as the hematopathologist and coagulation and also point of care. So Dr. Tariq Aweda is currently consultant hematologist and transfusion medicine at King Faisal Specialist Hospital and Research Center. He is the professor of hematology and transfusion medicine at Al Faisal University, Riyadh. And also he previously as the executive medical director of the Al Habib Medical Group Laboratories in Saudi Arabia and Gulf region. Also, he graduated and obtained as the MBBS at the King Saud University in 1993. He received his FRC bath at American College of Physicians, Philadelphia, USA in 2009. He also a member of Saudi Point of Care Testing Association and POCT, and he published several papers in different journals, national and international. And also he received several awards for best research and best publication. And also I think as a best teacher and trainer. On behalf of all the audience, I would like to welcome Dr. Tariq Aweda, and I will ask him to start his talk. Please, Dr. Aweda.
Assalamu alaikum jami'an. Good evening, everybody. Shukran, Walid. It's my pleasure to be with the Saudi Society for Clinical Chemistry and to talk about subject that I think shared by chemistry, hematology, and many other specialties. I'll try today, inshallah, to go through the coagulation and point of care in coagulation. If you allow me just to share the slides. I hope, is it clear, Walid, the slide? I can start. You can see the slides?
Clear, yes.
Okay. So coagulation and point of care, it's a challenge, it's a future, and it has a lot of things to do with different players in the field of coagulation and point of care. I will start just having some introduction about point of care in general, and then I will spend some time just to introduce you to the coagulation and a complex system, then goes through some of the available coagulation tests that we practice in both central laboratories and at different locations of point of care. Then I spend a little bit of time talking about point of care for antiplatelet, which is related to the subject of coagulation. Then before ending the talk, I'll go through the home monitoring self-testing using self-testing point of care and how we can utilize the connectivity to join all these surfaces to produce better clinical outcome. So let's start with the point of care. What is the point of care? We all know, and the field, it's a point of care testing that is near patient testing, or the test that is done outside the medical laboratories, and usually it is done by non-medical technologists, mainly nurses or physicians, but sometimes other medical providers can provide, including, as we will talk later, the patient themselves can be using part of the point of care, which is a home self-testing instruments. The point of care can be in anywhere. It can be at hospitals, can be near patients, can be sometimes in labs. Some of the labs, they use point of care equipment to deliver quick services. And the best example now, what we hear and see with the COVID, where point of care testing for COVID test is available in some of the labs to produce the quick result within short time. The purpose of the point of care is to improve the patient outcome. So point of care provide us with reduced time to results, which we need to improve patient outcome. It reduced time to treatment. So we initiate the treatment earlier. We adjust the treatment if there is a need before we wait to go for labs for other things before or that may delay the treatment. So it reduces the time to the treatment. The change of the treatment would be easier, and we will talk a little bit more about the change in the treatment when we come to the home monitoring or change of the treatment or change of the dosing in some of the location in the hospital. The point of care not only help in the clinical side, but also it would help the hospital general or central laboratory by reducing unnecessarily testing. The best example is the point of care that can be done in urinalysis, where if you do the dipstick and in a point of care, you don't need to send a sample if your point of care is well organized, well controlled. Same for INR, same for other laboratories. So it reduced the duplication, reduced the load on the laboratory testing, reduce unnecessarily prescription, especially the antibiotic, or when there is a point of care that can help you to identify things like strep A or some of the infections or some of the laboratory or some of the clinical situation where the point of care can give you the test. It will reduce the prescription. And the other example is the D-dimer. Using the D-dimer at point of care in the ER can reduce the unnecessarily movement for treatment with anticoagulation. It reduces clinical visits, and this is quite obviously seen with the home monitoring, but it's the same if you apply the point of care. And again, talking about anticoagulation clinic, if you have a point of care in the anticoagulation clinic, that would minimize the visits of the patient to the hospitals, because the patient can come one time instead of coming twice, one time for the testing and one time for the visit to the clinical service. And it can reduce the referral if you have outreach program where you can provide the outreach or remote clinics, or even family clinics with the point of care. They can do it there and reduce. And the same can be done with the endoscopy using H. pylori near the patient. All this would result in reduced length of stay at hospital at any point, and as a consequence, there is a clinical outcome that would be measurable and that will be improved. If I take you to the coagulation is a complex system, and it's maybe one of the most complex system in the body, where many players, including the endothelial cells in the vascular system, different blood cells, not only the platelet, but even the red blood cells and white blood cells are important for the different parts of the coagulation. It starts definitely with the platelet shape changes and release to close any gap that happens or any defect in the endothelial, aggregation of the platelet, then formation of the platelet clump. And as a consequence of the formation of the platelet clumps, you need to go to the next step, which is the clot formation and fibrin formation, which will trigger the next step, which all happens many times instantly on the same time, on the same cells, where both the extrinsic and intrinsic pathway using different proteins and factors would take care of the cementing of the defect that happens in the vascular system, and result as an end result of formation of fibrin glue or fibrin monomers that will seal the things and stop the bleeding. This all can be complex and can be tested by many tests. These tests can be either tests done at the lab or at the point of care. But we have to understand that the test initially, when it was formed, or most of the coagulation test was designed for measurements of protein cascade, the cascade I just showed you, to identify if there is a shortage or deficiency or absence of one of these proteins. And the most common lab tests that we are used for routine practice are the activated partial thrombin time, the prothrombin time, and thrombin time. And adding to that, another test which is not available in the central laboratory, but it is with its start, it was initiated in the point of care and continue to be the point of care. And we will talk a little bit more about the activated clotting time, its value and use. We have to remember that they don't mimic in vivo process. And they are designed to detect the abnormalities in the proteins and hepatic failures. But we found that it can be used for monitoring different anticoagulants, particularly heparin and warfarin. And that was long time, more than 50 years ago. The coagulation test can be divided according to the cascade. So it helps us to identify problems that may happen in the intrinsic pathway. And the best tests to use are the ABTT in the lab and point of care, and ACT on the point of care setting. For the extrinsic pathway, we use DBT, INR, for measurements of defects that happens with the point of care or defects happens with the extrinsic pathway. And if there are defects outside these sites, we can use another two tests, which are the thrombin time and the fibrinogen test, to identify problems that may happen in the common pathway. As I told you before, we can divide the groups of coagulation tests to two settings. The setting of a routine lab or specialized lab, where we can use ABTT, BT, D-dimer, thrombin time, fibrinogen, and DUAX, which can be shared with the point of care, because all these tests can be done on both sides, or specialized tests that can be done only on one side, which are the anti-10A, anti-2, or different factor assays. On the other side, ACT and Thromboelestography are particularly point of care. So these are the group of tests. Some are shared by both, and some are designed to do on either sides. What are the difference between these settings? Simply, the standard laboratory use platelet poor plasma, so you need to use an anticoagulated collected samples, collected in sodium citrate in a special collection, special dilution, where in the point of care it is whole blood, no anticoagulants needed to be added, and no dilutions need to be added, so it's ready to be used right away. There are variable pre-analytical delay because of transportation, because of phlebotomists. This is not available in the or not seen in the setting of point of care, but we have both sides share the pre-analytical error, which includes operator and quality, and all measures related to the sample quality and operator of the test, and they share these errors that we need to take care and optimize it before we get the results. There are different point of care coagulation analyzers. Different companies produce different instruments. This is just a list of some of these equipments. We get frequently updated a list of these tests, and these tests have different menu, different sizes, and different cartilage, and it can be done by different complexity. Some are very easy to be used, some are more complex and needs more training. But what we need these tests for, we need them for the mainly not for making the diagnosis of problems that happens with coagulation from deficiencies or problem with the bleeding, but we need them mainly for monitoring anticoagulant drugs. For unfractionated heparin, ABTT is the test of choice, but ACT can be used in setting of point of care. We'll talk a little bit more about it. And if we have problems with ABTT or ACT, we have the more gold standard test, the heparin assay, or the anti-10A or titrated heparin tests that are available, mainly available in the lab. Warfarin is the second and most commonly used drug, until recently, when other drugs came to the market. It used to be there for more than 50 years. We left with the warfarin, we left with its limitations, its advantages and disadvantages, and until now, warfarin continue to be the most commonly prescribed for medical different medical indications. And for monitoring warfarin, we use the BT-INR, and that's the standard way with a standard method with a standard intensity. Depends on the diagnosis and the need for the warfarin. Low molecular weight heparin, it's the most commonly prescribed in hospital and out hospital, and usually doesn't need to be monitored, but in a special situation, a special clinical condition, you need to monitor if the patient is responding or the patient have side effects of the drug. So we use anti-10A chromogenic assay. The problem with the anti-chromogenic assay is mainly located in the lab. Now with the need to have the antitin A, several companies are working to move the antitin A from the lab to point of care, where it will be easy to adopt it for adjustment of the patient drug level for patients using low molecular weight. For the last 10 years, we have a new group of drugs called direct oral anticoagulants, and now currently we have at least 5 approved types of direct oral anticoagulants. Again, these drugs usually they don't need to be monitored, and they are coming quite fast to replace warfarin in many indications. And when there is a need for test, we have an antitin A calibrated chromogenic assay, and again that test is available in the main routine lab, but again several companies and some of them are close to release the test or point of care test for DUACs. I'll go quickly through some of these lab tests that just elaborate about their usefulness, especially at the point of care. We said ABTT is mainly important in 2 areas, the intrinsic and the common pathway, and it measures by automated system using an activator like choline or citrate, and it can detect the congenital abnormalities or defects in the different factors, either by acquired antibodies or congenitally defected factors. It can detect presence of lupus antibodies by getting elevated, presence of factor-specific antibodies, and it is important in measurements the effect of heparin and can be elevated in patients with warfarin. It can be affected by many factors, the system itself, the reagent, and presence of heparin at different levels, be it low and moderate. And putting in mind the heparin at very high levels, it's not good test for. So ABTT shouldn't be used for high heparin. Those who receive heparin dose of more than 50 IU per kg shouldn't use this one, and that's where ACT can be the test of choice. The ACT resemble the ABTT, but it's not equivalent to ABTT. It detects the same, the intrinsic pathway and the part of the extrinsic part of the common pathway, but it, as I mentioned, it is mainly a point of care test. It's not available in the lab, and there is no equivalent test to this, and it shouldn't be treated as a BTT and shouldn't be validated against a BTT because it gives different results. It used the same choline or ciliate or glass beads as an activator, but with higher concentration, and it lacks complete correlation with the ABTT or antitin A, and it is very good for monitoring patients on heparin where the ABTT lose its specificity and sensitivity and become useless, and has different response to the trace amount of heparin, but generally those who receive heparin at low dose or moderate dose should use ABTT. The activated clotting time was majorly designed to monitor heparin in the high concentration. Some of the cartilage can detect the moderate level, but it's not advisable. It is not a predictor of bleeding, so we should not use the ACT to predict if the patient bleeds or not. And that by mistake had been used in ECMO patients. And recently we had published in the pharmacology journal that showing that ACT has no value to predict those patients who bleeds. So if you suspect patient with bleed and he's or she's on ECMO, you need to use antitin A instead of ACT. There are different criteria needs to be met before you use the ACT for monitoring. You should have a normal antithrombin 3 and normal factor 12 level. Platelet should be above 50,000. The patient has to be normothermic because that affects the result, and the hematocrit should be within acceptable range, above 0.28 or 28%. That's the time when the tests become more measurable and acceptable. There are different things can affect, again, the ACT as in ABTT. The method of testing, if you are not using a standard method of testing or the method was not calibrated, presence of lupus anticoagulants, if the patient has a factor deficiency that affect the level, patient with hypothermia or hemodilution, or the platelet count less than 50, that affect the activated clotting time and may give wrong results. And presence of drugs like R-protein can affect the measurements of ACT. Different, again, coagulometers are available for measurements of ACT, and each one has its own cons and pros. And if you are using any one of these, you need to know the limitations and how you use it before introducing this instrument to your clinical service. There are many clinical evidence that looked at the ACT and ABTT and in different settings like ICU, the BCI in coagulation, and in the setting of cardiac surgery or neuroangiographic surgery. And definitely all of them had shown that the ACT is the method of choice for monitoring the anticoagulation in the high concentration heparin, but not in the low concentration heparin. How to overcome the problems that may arise with the ACT. Standardization of the instrument, procedure, and reporting is very important initial step to get a good ACT results. Good quality control, which can be electronic, liquid, using external, and the guidelines for performance is very important. So particularly, you know all that the ACT will be used by nurses and by physicians who have less experience when it comes to the quality control and dealing with instruments. So putting guidelines that would help the operators to perform the test in a good manner would result in improvement of the results and decrease the problems that may arise with using these instruments. Serial measurement should be monitored instead of individualized clotting time values, and you need to have auditing from time to time to be sure that the ACT is run in a good quality manner. ACT can be used in cardiac surgery, interventional surgery, different critical care settings, and different other satellite, the dialysis, ECMO, emergency, and anticoagulation clinic if you have patients who are coming with high doses or use high doses, moderate to high doses heparin infusion in a setting of a hospital. Prothrombin time is the third type of test. It's basically used for monitoring warfarin therapy and monitor the warfarin by using international or INR, international normalized ratio, at different target level, be it prophylactic or therapeutic, be it on artificial heart valve or due to thrombosis. And the use of the us to adjust the dosing total patient management if it's done within a logarithm, within a setting that address the results of the point of care. D-dimer is the last test I will just highlight on it, because D-dimer is introduced as a negative predictive test for emergency and for critical care, where there is a suspicion of a DVT. Run the D-dimer. If D-dimer is negative, you can stop and don't take the patient to radiology or ultrasound. The use of different D-dimer assay shows different accuracies. So you need to know your accuracy. If it is high accuracy, high negative predictive value, you can use it, and you may avoid using diagnostic radiological test, and you can release the patient safely. But you have to be sure that before introducing the D-dimer as a negative predictor value for your laboratory, for your clinical setting, you need to be sure that this is run in a good quality and it has a sensitivity and a negative predictive value that is acceptable. Thromboelestography is another test that tests the whole blood. It's a global test for testing the whole scenario of blood formation. It uses a small amount of blood, and it is used mainly for surgery, complex surgery like liver and cardiac, where coagulation defects can happen due to changes in the cardiac and changes in the platelet. So the test can be used to detect both bleeding and coagulation, and it can help in guidance of transfusion requirements. This sensitivity and specificity had been proven for preventing the bleeding and guiding the transfusion. ROTEM is one of the methods used, the TEG principle, and because of the time, just I will pass quickly. It used the impact of the change in the rotation of the a wire that will be changed as a clot formation, and it will give us a graph, and that graph can guide us about the coagulation system in the patient. Monitoring of antiplatelet can happen with many equipments, and it's at one point was one of the tests that been promoted to monitor patients using antiplatelet, be it aspirin or Plavix. Now the demand to go for that is less, and only it's been recommended for patients showing antiplatelet resistance. Many systems are available for testing the platelets, from BFA, which is commonly used, to multiplate, platelet aggregation, Verify 1, and even TIC can be used. The new TICs can be used for that. Before ending my talk, just I would like to share with you the home monitoring principle, which is an important monitoring surface that will help you to decrease the frequency of texting, maintain therapeutic range for patients using warfarin, and improve the whole health status of the patient by decreasing the patient visit to the clinic, and improve the clinical care at home setting. It is an effective tool to monitor the patient compliance to the treatment, where the patient can do a weekly instead of monthly testing. And we had shown, and many studies had shown, that if you test the patient or monitor the patient on warfarin on regularly, more regularly than monthly, you will be getting better patient in time, or time and range, where the patient is adequately monitored and adequately in therapeutic range. This is a summary of meta-analysis done in different setting, different situation, and clearly you can see that all favors the use of home monitoring devices to adjust or to monitor patients who are using warfarin. Benefits, including creating a greater time in therapeutic range, as been shown by many studies. Fewer dosage changes, because as you monitor the patient, you will most of the time don't need to make a change. It reduces the complication of using of warfarin by reducing bleeding and thrombotic complication. improve overall survival, improve the quality of life of the patient, because the patient doesn't need to come frequently. Patient can come every 2 to 3 months instead of coming every month, and shorter than every month if the patient is not on therapeutic, and definitely significantly cost saving. There are many studies, again, because of the sake of the time, I will not be going through, but you can see, although we know that the immediate cost of the point of care test is high, but the total cost, if you look at it compared to the prothrombin level, you will be getting a less or at least equivalent total cost for measurements of INR at home. If again use it in more sophisticated computer-based management, you may improve the patient care better, reduce the need for physicians, and adjust the level of the INR more precisely. Just quick snapshot on one of the exercise we did at King Faisal 10 years ago, actually, not recently, where we looked at the use of INR at home using a method of choice for patients or selected patients, where we give them the instrument and they did weekly for 6 weeks reading. And as you can see, this is a snapshot of the 9 patients where with using the instrument at home, we managed to keep the patients ranging between 50 to 100%. Some of the patients had a difficulty, like this patient and this patient, where they had a difficulty to complete the study or to have adequate results. But in 80% of our cohort, the patients managed to have it at least 50%, and some were getting results almost 100% compared to the central lab. And this is reflecting the R283, comparing both the results of the lab and the INR measured at home. Connectivity is important, and I will end my talk just by highlighting that the connectivity for point of care, particularly for coagulation, is very important, because it will enable the point of care coordinator to monitor the instruments remotely, and at the same time it will enable the instruments to interface with the hospital information or lab information system, and that will keep the results safe and have a full journey of the patient during his visit to the different sites of the hospital, or during his testing using the central laboratory testing. By that, I would like to stop here and thank you for listening. Dr. Walid?
Okay, Assalamu alaikum. Thank you, Dr. Tariq, for your good overview about the point of care testing and at the same time coagulation. So to join 2 important topics and in one talk, this is usually done by expert, what you call, doctor like you, Dr. Tariq. So we'll keep the results, keep the question at the end of this seminar. Now we'll go to the second speaker for our seminar, Dr. Malak Al-Mish'ali. She will talk about the role of INR point of care testing in the time of the COVID. Mish'ali, she is the consultant clinical scientist, point of care in charge at Prince Sultan Military Medical City, Central Military Laboratory and Blood Bank, Riyadh. She obtained her bachelor degree in applied medical sciences, medical laboratory at King Saud University in Saudi Arabia in 2005, and received her master degree in biomedical science, clinical chemistry, point of care testing in 2012, and she got also the PhD degree in point of care testing in 2018 at Manchester Metropolitan University. Also, she's a member of POCT, and also she has published and presented several papers in different journals, national and international. I would like to welcome Dr. Malak, and I would also like to
thank you, Dr. Walid. Thank you, Dr. Walid. الصوت واضح؟
أيوة واضح دكتور.
Assalamu alaikum wa rahmatullah. First, I would like to thank the organizer of this meeting for inviting me here today. We all know that we are facing this tremendous problem of COVID-19 disease since the first case discovered in December 2019 in China, Wuhan. And since then, there is a lot of innovative changes has been introduced to the clinical practice. My talk today is particularly related to the implementation of the INR point-of-care testing in coagulation clinic in highest quality management system, and to put the insight for some recommendation and innovative strategies might help during such a situation. So our objective today is to understand the role of point-of-care testing in public health, and to understand the basic criteria for selecting POCT PTINR device in the coagulation clinic, and to understand the implementation requirement in terms of quality and safety in coagulation clinic. And finally, we'll talk about the pandemic, and sharing with you some innovative strategies has been implemented internationally. So what is the point-of-care testing? The point-of-care testing is the bedside devices where the healthcare providers having their result at the patient side within a few minutes. And as you can see here in the slide, there is several POCT devices in the market, which include but not limited to the cardiac panel, the coagulation profile, the glucometer, the urine dipstick, in addition to the blood gas analyzer. So the main function of the point-of-care testing is faster turnaround time, where the doctors can having the result quicker, which will improve the immediate patient management and accelerate the decision that will ensure better clinical outcome and improve patient satisfaction. I will start with the definition of the Prothrombin time. The Prothrombin assay is the most requested coagulation test used for monitoring vitamin K antagonist therapy, where the result may expressed as international normalized ratio, INR. It is vary with the type of thromboplastin, which I will talk about this later in this presentation. The differences in sensitivity among the assay or the method is known as sensitivity index, and the INR is a patient prothrombin time, which mathematically adjusted, as you can see here in the equation in the slide. And the INR is developed to standardize the PT results between different PT analyzers and thromboplastin reagents. So several studies have classified the point-of-care INR for anticoagulation clinic as a reliable and rapid alternative assessment than the laboratory standard test. It has been approved that the INR POCT test enhancing patient satisfaction with significant improvement in waiting time. Would take into consideration the disagreement and discrepancy between POCT INR and the standard laboratory INR, which I will explain this also later in this presentation. In this slide, I'm sharing with you the benefits and the advantages of implementing INR test in the clinical practice. First, with patient finger prick sample, it will decrease the need of the venipuncture test, so there is no need to collect a blood sample. Also, patient on anticoagulation therapy need to maintain a careful balance between their clotting and bleeding risk, ensuring that their INR falls within the therapeutic range. This will avoid the risk of hemorrhage. It's also simplify the workflow at the coagulation clinic and enhancing patient satisfaction with reduction of waiting time. And finally, in terms of the cost, it will decrease of long-term anticoagulation therapy as studied showed. I will share with you here some important selection criteria for choosing the INR point of care instrument, as there's several types in the market. First, it's important to check the analytical sensitivity of the device that meet the quality assurance of your hospital. It's important to ensure that the device you select having a quality control and external quality assessment are available. Also, in terms of the cost, evaluating the total cost of the implementation process, which will include the consumables that you are going to use, the quality control, the quality assurance or proficiency testing program. And finally, it is important to check the manufacturer insert sheet for any factors that can interface with the point of care instrument. For example, the presence of the antiphospholipid antibodies, or a low molecular weight of heparin, and also in severe anemic patients, as it will lead to elevate the INR values. The sample type and the finger pricks. What is the sample type for the POCT? It's usually the capillary whole blood from the finger prick or venous whole blood, anticoagulant-free blood tube, citrated whole blood. And when choosing the finger prick, it's very important to consider the length and the depth which compatible with the POCT instrument that you select, and it's also be convenient to the patient with the less painful type. So now, after you selecting the device and before the implementation, it's important to evaluate the device and ensure the validity of the device in terms of precision, accuracy, and also correlation to the laboratory method. Ensure that the device is meeting the quality standards in the hospital if the hospital is accredited by external bodies like College of American Pathology, SBAHI, JCI, or ISO. After the method validation process, it is essential to design a training program for all POCT users and selecting a trainer with having a clear education material explaining the policy and procedure which will involve the user should know how to run the sample, what is the documentation needed, where to document the results, where to document the quality control, and how to report the result as well. Some devices in the market comes with operator lockout features where only trained and competent users are eligible to run the sample. So it will be locked if the operator ID is expired and they can't run the test. Also, the patient lockout feature is important to avoid the using of random numbers and to comply with International Patient Safety Goal number 1 to identify patient correctly. So as we know that the test process can be divided into 3 different stages: the pre-analytical, analytical, and post-analytical stage. During the training day, ensure that the POCT users are aware of all of these stages. For example, in the pre-analytical stage, users should be aware about the patient factor that include the patient diet or medication, also the specimen collection tube, the site, the preparation and storage of the lancet or the cuvette, and the quality control. In the analytical stage, to know the type of the thromboplastin and the ISI calibration, also the factor affecting the assay sensitivity. In the post-analytical stage, a user should be aware about the INR calculation and the correct way of result reporting. So to select a machine, ask the vendor about the availability to connect the device to the laboratory information system or the hospital information system through a middleware. As the connectivity is very important, so the users can, the POCT coordinator from the lab, they can check the user's activity and they can check the quality control to ensure the regulatory compliance with effective management in place. In terms of the cost, before considering in terms of the cost for look at the whole process of the patient care rather than the cost of the individual assay versus the cost of the laboratory. Evaluate the cost of the training, the consumables, the proficiency testing program. However, several studies has been shown that the POCT INR test is cost effective when considering indirect expenses of the hospital staff, resources utilization, and management of adverse event. In this slide, I will share with you the differences between the point of care testing and the laboratory test. The sample using in the POCT test is the finger stick whole blood sample, and the lab, they're using citrated plasma derived from the venipuncture. The thromboplastin in POCT test is a different type, and in the lab, it's usually human thromboplastin. The method of the endpoint detection in POCT, electrochemical sensor mechanical method, and in the lab, photo-optical mechanical. The ISI and the main normal prothrombin time in the POCT test is manufacturer determined, or and on the other hand, in the laboratory test, it's local calibration. The availability of the patient self-testing, it's available in the POCT test and patient self-management, but it's not available in the laboratory test. Here is the most common errors related to the point of care in coagulation clinic. The preparation time or the pre-analytical error, as I mentioned earlier in the presentation, the reliability of the POCT instrument and medical condition of the patient, also inaccurate POCT INR result may increase the risk of bleeding and clotting. Taking the finger prick sample from unwashed hand will affect the accuracy of the result. Also, it is important to not only report the verified result with the patient to avoid the patient anxiety for the waiting time. So, for example, if you're having POCT INR results above 4, and you need to send a confirmatory sample to the lab. So share with the patient only the verified result from the lab to avoid the anxiety of the patient. In this slide, I'm sharing with you a scenario. I'm pretty sure that most of us experience this scenario in daily basis. The POCT user, the nurses, phoned the POCT lab complaining about the discrepancy of the result between the POCT and the laboratory result. The clinical pharmacist asking why the INR results are so different and which results should be used to guide the warfarin dosing. Doctors asking for confirmatory test, sending another sample to the lab. The point of care technologist explaining to the user the reason of the differences in the result and why there is a discrepancy between the POCT and the lab, and the patient complaining about the result and say, I don't trust this device, or which increased anxiety and waiting time for the patient. So this leads directly to my next point here about the reason of the variability of the POCT INR versus the lab standards method, where using a different type of the reagent, different thromboplastin and different lot number also of the reagent with different ISI calculation, As I mentioned before, that the POCT device are usually, the ISI in POCT device usually set by the manufacturer. This is a call to standardize across different manufacturers and reagent, so it can be anticipated for more accurate results between different analyzers. There is some limitation of the point-of-care INR test, where similar studies indicate that point-of-care instrument showed a disagree or bias with a result greater than 3.5. However, caution is needed with regard to higher INR result, which call for laboratory confirmation test. I'm sharing with you here a quality and safety consideration tips. So with the INR above 4, it cannot be compared to the lab result due to a big margin and discrepancy, subjected to a confirmation sample to be sent to the lab. If the machine accepted by the clinical, it's important to work closely with the laboratory to ensure the correct calculation and to be aware about the ISI calculation in the lab with the strict policy, and to evaluate, the evaluation should be closely related to the clinical issue. Also, it's important to have a quality management system in place and to participate in proficiency testing scheme to ensure the accuracy of the results during the year. The awareness of the disagreement between the lab and POCT result, and also to have a well-designed training program for all the users in the hospital. And finally, auditing. So having a monthly auditing to ensure the quality of the work and documentation following the policy and the procedure of the hospital. So here I'm reaching to the second part of the presentation. Since the coronavirus COVID-19 disease has been started, the World Health Organization recommend to stay home, maintain social distancing, and stay away from those who are sick or could be infected. However, during the pandemic, several measures has been implemented at the hospital to minimize the exposure of patient and healthcare providers to the virus. So the pandemic forced to reshape of the clinical practice and give the rise to the need of innovative solutions that would maintain standards of the patient despite the physical distancing. Let me now turn to the importance of keeping anticoagulant service flowing during the COVID-19 pandemic, as the anticoagulation drug has been classified as a high-risk medication. Having a practical guidance has, several practical guidance has been implemented in several hospitals around the world to ensure that anticoagulation care continue with safe practice to ensure the patient safety as well. So this brings us to the suggested action and measures can be implemented in the hospital to check the patient symptom or confirm COVID-19 infection patient, review the patient on individual basis, also perform an assessment of a risk inside the clinic, and careful measures of the hand hygiene and personal protective equipment for both the patients and POCT user. So what can anticoagulation providers do to minimize the exposure risks for our patient? These are several strategies has been implemented in several practice internationally with good impact to patient safety as published. For example, scheduling a strict appointment agenda to minimize the social distancing, Also, anticoagulation hotline available for the oral anticoagulant patient, so they can ask and share their concern with healthcare providers. In addition to that, some practice introduce the telehealth consultation, where the patient also can contact their doctors directly and the clinical pharmacist regarding their condition and medication dose. Also, some practice implement a drive-through where the patient can having their result and test a sample on the site in safe environment. And finally, some hospitals assess candidacy for self-management or self-patient testing of monitoring the INR. Let's take a look at the self-testing using the point-of-care device. The self-monitoring with the coagulometer may reduce the frequency of visits to the hospital or the clinic for the patient and enable them to be monitored more regularly. And here, there's some benefits of the home monitoring, having immediate warning if the patient testing was done improperly. It is easy maintenance, and device is easy to learn also and to use. It's only require a finger stick instead of a full blood sample. It's very convenient and limit the frequent visit to the lab. Also, it is a simple reporting of the result by the phone to their doctors or healthcare providers. The disadvantages of the home testing, that the home INR don't work very well for the people with antiphospholipid syndrome, and it may not work well for the patients with INR above 4. So where do we go from here? The important thing about good and safe clinical practice in a coagulation clinic is good communication and collaboration among all the point of care coagulation stakeholders, including the laboratorian, the doctors, the clinical pharmacists, and the device providers. And finally, I will end here with some recommendation might lead to safe practice for implementing the patient self-testing in clinical practice in such a situation during the pandemic. First, having a clinical effectiveness of patient self-testing among Saudi population should be implemented and evaluated. Also, national guidelines and recommendation for patient self-testing must be introduced. Having an economic analysis to identify the cost for all stages. Carefully selected patient criteria for patient self-testing and patient self-management. Design a clear education material for patient self-testing and patient self-management and their caregiver. It's needed an engagement and effort for all the multidisciplinary team and high quality anticoagulation management committee to set the guidelines and to evaluate the risk to ensure the patient safety. And finally, innovative strategies that lead to long-lasting change in the coming years. I'm sharing with you here in the slides some key documents to ensure the highest quality and to improve the INR POCT service in the coagulation clinic. A document from the College of American Pathology regarding the point of care testing for coagulation, limitation and challenges, and another document from the Clinical Laboratory Standard Institute, and finally the guidelines from the Anticoagulation Forum for managing anticoagulation during COVID-19, published 2020. Thank you for joining us today, and I would be happy to answer all your questions at the end of this seminar. Thank you.
Okay, thank you, Dr. Malak, for your talk and also to explain the total testing process, also for point of care take time for explain the pre-analytical and analytical post-analytical issue with the point of care testing for the coagulation, and also to cover the topic how to use the point of care testing on the COVID-19 era. Now, I'll just to remind the attendee about by attending this seminar, you will get an A in the 3 CME, but you should at the end of the seminar to complete the questionnaire. Okay. After the completing the questionnaire, you will be, inshallah, receive the 3 CME for this event. The last talk today with Mr. Rani Besiso from ROCHE Diagnostic. Mr. Rani, he's currently the Medical Affairs Manager at ROCHE Diagnostic in Saudi Arabia. He previously the sales manager at ROCHE Diagnostic between 2017 and 2018. He obtained his bachelor degree in pharmacy at Amman Private University. He has received honors and awards, including the top 3 management ROCHE Global, KSA Success Stories, TLA Project Award for 3 consecutive years. Pegasus Product Champion in 2004, runner-up man for the year in ROCHE Saudi Arabia in 2002, and Roseven and Tamiflu Product Champion in 2001. Mr. Rani, he will present a talk about the introduction to the Prothrombin Time and its various application from a diagnostic point of view. So I would like to ask Mr. Rani to join us and to present his talk. And also I will remind the attendee to send their question to the chat to prepare the question for the speaker at the end of this seminar. And thank you. Please, Mr. Rani.
Thank you so much, Dr. Walid. Good evening. It's really a pleasure being in this distinguished educational event. Special thanks to Saudi Society of Clinical Chemistry and respected speaker, Dr. Tariq and Dr. Malak, and with the whole audience. Actually, I will go just quick through about, in the coming 25 minutes, about the introduction of Prothrombin Time and its various application from diagnostic perspective. So I will just go about a quick overview about Prothrombin from history and to other standardization. Also, we'll talk about the anticoagulant clinics and the specialty. Sorry, I will go for Prothrombin Time and we'll go for anticoagulant clinic and site testing for patient self-testing, and then we'll go for a simple summary. So Prothrombin Time, it as a history was the first described and published in 1935 by Mr. A.J. Quick. It was really observed that coagulation time depends on the amount of thrombin when calcium ions and tissue extract are added to critical plasma, to saturated plasma, sorry. And the nomenclature of Prothrombin Time is a sort of misleading as it's oversimplified the test. However, with regard to its dependence and interchanging thromboplastins. Therefore, if we just like to go to the test principle about the Prothrombin Time, we'll be able to find that the assay contains thromboplastin and calcium, in which they initiate the activation of the extrinsic coagulation cascade when added to saturated human plasma, as we can see on the right hand of the slide. Thromboplastin as a tissue extract containing abundance of tissue factor and coagulation activation phospholipids. Therefore, PT or Prothrombin Time can be considered as a global coagulation test, and it comprised several enzymatic reactions. Prothrombin time. The test principle is elapsed between the addition of the reagent to the plasma and the formation of the fibrin clot. It measured and reported in different format, seconds, percentage of normal, and INR. Calibration is not applicable for primary units, second, but it's available for INR and percentage, as we all know. But when we go for prothrombin time as a PT, INR, and percentage calibration, the routine calibrators are traceable to the WHO standards, so they are referred to the WHO standard. INR based on INR calibration with the prothrombin calibrators set. The percentage based on the percentage calibration with the PT calibrator set. For prothrombin time recognition, which is ROCHE Diagnostic provided, the conversion of patient prothrombin time to INR is based on International Sensitive Index and mean normal prothrombin time. So it's revered and standardized. Now, when we go to prothrombin time, how to get from second into percentage. Prothrombin time is very intuitive test, diluting pooled normal plasma with 100% activity. And therefore, the plots of paired data, the percentage of activities versus clotting time, allow the preparation of the calibration curve that can be used to interpolate the percentage activity from patient prothrombin. Now, prothrombin time, how to get from second into percentage. The expression in percentage has some limitation. The relationship between the clotting times and the percentage activity is not linear, unless it has been modified, for example, like log scale applied to the data. And therefore, we can say the small clotting time variation in healthy subject, which will lead to large variation of percentage activity, leading to values for higher than 100%. And therefore, the potential, there will be a potential for incorrect interpretation as hypercoagulability. Now, the limitation with seconds and percentage. There is lack of standardization, why values are not comparable between laboratories. Same point that raised by Dr. Malak. So calibration of the thromboplastin with different calibration plasma, the influence of the different measuring devices on the results, as there is no uniformity, individual sensitivities of different thromboplastin vendors and different factors lead to result obtained with different type of thromboplastin are not comparable. And the anticoagulation effect as well is also not comparable. So how to overcome this issue? With INR, an attempt to standardize anticoagulation monitoring, which is introduced by the WHO in 1983, allows the comparison of prothrombin values used to control oral anticoagulant therapy with vitamin K antagonist, validate the stable phase for anticoagulant therapy, and the accuracy of INR system is only defined, as mentioned by the previous speaker, for a range between 1.5 to maximum 4.5. So how to get from seconds to INR? So the International Sensitive Index enable results to be obtained that are independent to the reagent. Specific thromboplastin reagent can be calibrated against WHO International Reference Standard. The mean normal prothrombin time is a geometric mean of the prothrombin time test in seconds of more than 20 healthy donors to be calibrated there. Now, if we'd like to talk about the ISI and the mean normal prothrombin time, often the manufacturer assign an international sensitive index, which may value as the value, but should verify it locally, where the interim mean normal prothrombin time value are not generally assigned. Laboratories are usually required to determine a local mean normal prothrombin time, and this can be accomplished by having at least 20 normal individual samples required to be calculated the geometric mean that will be identified. So prothrombin time, how the International Sensitive Index is obtained. Actually, this is a very important slide because when we are talking about prothrombin time, systems are based on 3 different species of thromboplastin: human, bovine, and rabbit. And in 2016, there are 2 references of both human-based thromboplastin and rabbit-based thromboplastin were released. Human-based thromboplastins and rabbit-based thromboplastin do not measure equally. Therefore, it's very important to know and to be aware of the type of thromboplastin used in the calibration when we use test strip or in central laboratory. The WHO expert committee recommends that commercial rabbit thromboplastin shall be calibrated with the international standard for rabbit thromboplastin and commercial human. Thromboplastins with international standards for human thromboplastin are very important to be standardized as and calibrated on the standard. Now, the international standard of IS, so the reference ranges for prothrombin is from 8.4 to 10 seconds. Percentage of normal is 74 to 120. The INR therapeutic range depends on the application area, like mechanical valves, atrial fibrillation, treatment of venous thrombosis, and INR from 0.5 to 1.9 low intensity, or from 2 to 3 conventional intensity. Now, we are talking about thromboplastin impact on different INRs. Thromboplastin reagent can vary significantly regarding their sensitivity and to reduction from level of vitamin K dependent clotting factors. And therefore, the recombinant thromboplastin are more sensitive to factor 7 than tissue extract thromboplastin. Therefore, the calibration of giving thromboplastin is more precise if performed against an international standard composition and from same species. Phospholipids and sodium chloride composition of recombinant thromboplastin are differentially influenced by relative sensitive to changes in the plasma level, especially of factors 5, 7, and 10. Now, prothrombin time when it's prolonged. As we know, vitamin K is needed for biosynthesis of all proenzymes of the serine proteases of prothrombin complex with the factors 2, 7, 9, and 10. Therefore, prothrombin time is usually and especially sensitive toward factor 7. Now, factor 2, factor 7, 9, and 10 actively levels can show variable reduce values. The first clotting factor usually assessed is factor 7 because it has a very short half-life. In vitamin K deficiency, prothrombin complex, as we can see on the right-hand side of the graph, prothrombin complex and protein S and C decreases, as well as in liver diseases conditions or disseminated intravascular coagulation, DIC, as well as factor 5 and antithrombin decreases, which allow differentiating the prothrombin time. The prothrombin time is still the most sensitive test for liver diseases, as we all know. It is not correct to think only about decreases of prothrombin complex when prothrombin is prolonged. There can be other reasons such as lack of factor 5, less influence but still, also inhibitors or hypo or dysfibrinogenemia. Now when we go about the application area, The main application area: monitoring of patient receiving vitamin K antagonist therapy, screening as for coagulopathies prior to any surgical intervention, detection of inherited or acquired factor deficiencies, part of ISSTH or DIC and SIC scoring system, liver function testing, and testing of vitamin K deficiency. Now, point of care INR versus laboratory INR. Which one to believe? And also this was really very well touched by Dr. Malak in the previous presentation. So the whole blood will better reflect in vivo hemostasis than testing with platelet poor plasma. The high quality anticoagulation management services typically know these risks and have SOPs to confirm elevated point of care INR results and monitor agreement between the clinical lab INR and point of care INR. Also, it is suggested that the case that an institution confirm all point of care INR results more than 5 in a chloride and bases all vitamin K antagonist adjustment on the INR values, although lab and point of care INR results have normally a good agreement. Potential disagreement between both was seen in trauma-induced coagulopathy. Also, it's very important in attempt to harmonize the INR results. Some anticoagulant management service have derived from INR-based management. So what we would like to consider in the INR, that the differences observed between different laboratories, different methodologies, whether they are using optical, mechanical detection, thromboplastins, point of care versus lab setting. INR system is defined for range from 1.5, 4.5 only. So there's non-linearity between systems with an I value outside this range. And focus should be on managing the patient, not managing the INR itself. So if we'd like to talk about the prothrombin time in summary. As a simple common hemostasis test, prothrombin time evaluates the extrinsic and common coagulopathy. Prothrombin time prolongation is indicated of a number of conditions, including vitamin K deficiency, liver disease, and DIC. It is used for vitamin K antagonist therapy, and the result sequence and percentage obtained with different thromboplastin are converted to INR for standardization. Limitation, actually, I believe the previous speakers were talking a lot about the limitation being not specific and different sensitivities, biological variation, and the elevation and risk. So here I would like also to stress on a very important point, which the anticoagulant clinics often report patient time and therapeutic range, which means to assess the anticoagulation management. Here, if you can see on the graph, that the time and therapeutic ranges, the higher that we have, or the higher that has been achieved, is more directly linked to a fewer adverse events in coagulation management. That's to say, the most recognized way to measure the therapeutic effectiveness of warfarin management, TTR or therapeutic time and range, has been shown to correlate strong with clinical outcome. And the laboratory should aim to hit more than 65% at least in TTR in order to minimize the adverse event on a patient level. Also a very important point for site testing, increased time in therapeutic range. Alternative site testing, more INR results are within the therapeutic range. Here, those studies are really evaluating the benefit of point of care testing with the decision to support the software and workflow design, and to see after implementing this approach, they found out the time in therapeutic range increased from 34 to almost double, which is 67%. And during 1 year of follow-up period, the complication related to anticoagulant therapy were reduced by 91%. And this reduction, I'm sure you all agree with me, that is really super major reduction in the coagulation, complication related point of view. Also, if we'd like to talk about the AST reduces the rate of complication. So the alternative site testing reduces the rate of complication for both thromboembolic and hemorrhagic events. How we can translate this? Actually, this is a review of 3 studies with more than 1,090 patients, and they found out that the AST achieved, the alternative site testing achieved 59% reduction in major hemorrhagic event and 68% reduction in thromboembolic events compared to usual care by using the alternative site testing or the patient self-testing. And this, and also, according to this, we expect that the alternative site testing reduces the rate of hospitalization. And also, those 2 clinical studies, separated studies, found that the usual care patient were 2.6 times and 4 times more likely to be hospitalized for warfarin-related complication rather than AST patients. Therefore, there will be reduction on the left-hand side study by almost 61% in the hospitalization rate versus the usual care. And on the right-hand side, we find 76% reduction in hospitalization by applying alternative site testing over the hospitalization setup. So if we'd like to conclude, realize significant benefit at every touch point, positive impact on clinician and patient. So the patient self-testing, there are multiple factors have been reduced for the favor of patients and the clinician, and increased also for the same thing. So the risk of transcription error was reduced, which lead to speed of data transmission and access for patient to medication, to management, and to better practice or quality of life. Turnaround time is just a brief definition about turnaround time is the time that the laboratory or the site can provide the result using the machine with a point of care or laboratory setup. So the turnaround time of the result were significantly reduced, and therefore the convenience of a treatment adjustment and management is increased because we get the result quite easily. The manual steps have been reduced, and there are almost 50% in the manual steps reduced from using site testing, alternative site testing over the hospitalization setup or the lab setup. Time saving for staff. I would also add time saving for staff, time saving for the clinician, and cost accordingly. Time on data entry, which lead to patient and clinician satisfaction. So that's the most important point of and by this I would like to finish my presentation, and I would like to thank you for your active listening, and I am happy to answer any question if there is. Please share it with us. Thank you.
Thank you, Mr. Rani, for your presentation. So now at the end of this seminar, I would like to ask the speaker to join, okay, to open their camera. Okay. I received some questions, and also I'm asking the audience and these to ask their question in the chat. Dr. Targa, whether about the normal I think it's this thing.
Can you repeat?
واضح?
الصوت بيقطع أبو وليد. Can you repeat again?
Okay. Regarding the hemodialysis, the normal level or the target level for INR for hemodialysis patient.
تمام. طبعا a hemodialysis patient who receive warfarin should be treated the same like others. يعني there is no special recommendation or special situation. If the patient is DVT and he's on warfarin for him and he is on dialysis, and for DVT, if the patient is not antiphospholipid, the standard is 2 to 3 INR. If he's the antiphospholipid with recurrence, the standard should be between 3 to 4, targeted 3.5. It depends on clinical situation, but renal dialysis doesn't affect the level of the measurements of or the intensity of INR.
Okay, thank you, Dr. Tariq. The second question for Dr. Malak about the role of the central lab to monitoring the point of care testing services. So the role of central lab to monitoring the point of care.
Well, the POCT service depends on the hospital regulation. Usually, the POCT service under the umbrella of the laboratory. So the POCT coordinator will oversight the quality control, because the POCT users usually they don't have the background of the laboratory. They don't know what is the quality control. They need training. They need the support from the lab. They need the lab. The support from the lab to ensure the quality regulatory for the quality control are in place. So the role of the lab is very important for the POCT, and usually the POCT devices is the evaluation of the method of the POCT devices from the lab side. Is this answer the question?
Okay, thank you. Yeah, okay, thank you, Dr. Malak. For Mr. Rani, one of the question: how can we use the plasma as a sample for point of care testing for the coagulation?
That's a very good question. Actually, we have the expert here, Dr. Tariq and Dr. Malak, but from the company point of view, yeah, if they allow me to answer, I will go. We
Definitely
use we use always the blood, the finger prick samples. And if they have more to add, actually, I would leave the stage for them. But
yeah,
our recommendation.
I agree. Yeah.
The plasma, they move to the central lab, and also we have also another giant analyzers there that they can support them.
Okay, Dr. Tariq.
وليد الصوت راح الصوت. Can you repeat again the question? راح الصوت.
بوينت في كيرة ضعيف الصوت. انواع النت جربتها ما ضبط معي.
طيب اتفضل الان.
طيب تسمعون الان؟
اي الان واضح، تفضل.
طيب هو السؤال عن الهوم monitoring for the coagulation by using air testing. What is the role of the hospital or central lab to monitor this the service, and how to let the patient to make a decision by himself to go to the emergency or to ask for medical advice?
تمام. It's a good question because home monitoring is an excellent service but very dangerous service. there is no standard of care that allow the patient to monitor by himself and adjust the dose. And the only ways to do that, he should or she should do it within a system that managed and controlled with the hospital. What I mean is that it's not as in the glucose where you can buy the small equipment for blood sugar where you can adjust your blood sugar or at least know how much your blood sugar, how much insulin you may need to receive. In coagulation, it's a bit more complex. Before using the home monitoring, you need to be trained. You need to have a criteria for that. You need to have somebody to monitor your performance, and you need to have from time to time adjustment or calibration with the main lab in the hospital. And for changing the doses or management of the dose of warfarin, it should be in cooperation with the anticoagulation clinic. And ideally, what is being done is you do the testing and you have the software, one of the most common software used internationally, like DAWN or like anticoagulation software, where the equipment interfaced or the home monitoring interfaced with this, and the result will be messaged to the physician or to the anticoagulation clinic operator, be it nurse or be it a pharmacist. And according to the advice, the patient will receive a message to tell him increase, decrease, or continue on your dose. So it shouldn't be a solo management, and it's quite dangerous if the patient buy the equipment and use it by him or herself without a system that control the home monitor.
Okay, thank you, Doctor. The question now to Dr. Malak regarding from your experience, Dr. Malak.
Yes.
Moni?
Yeah.
Now, from your experience, so what is the challenge for to get the false housings for the coagulation test? So it's related to mainly to the pre-analytical or analytical or post-analytical issue from your experience.
Well, I can share with you from a real practice. We did an investigation in the coagulation clinic because both the patient and the POCT users complaining about the results, and we found out most of the false negative or false positive from the pre-analytical stage. For example, it was a simple change improved the quality, the length and the depth of the finger prick has been using at that time. It's not compatible to the instrument. So the POCT users squeeze the finger to having more blood, and this will lead to increase the factors and to having inaccurate results. So once we change the finger prick to the suitable finger prick that compatible to the recommendation from the manufacturer, there's a massive improvement in the results. So the pre-analytical stages usually is most, and also the analytical. If there's an in-house ISI correction with the lab. As I mentioned that before, the ISI usually sit by the manufacturer in the POCT. So a training, well-designed training program is very important for the users.
Okay, thank you, Dr. Malak. Mr. Rani, just to explain for us about the role of IT and point of care testing, and what is the IT solution for point of care testing, especially for coagulation profile. أنا ما أسمع بس يمكن الصوت ما نسمع.
يمكن mute.
Yes, sorry.
تفضل.
So the IT solution is really very important for the point of care testing. However, if I would like to give more ideas about the IT, which is like the connected network in which enable patient physician to be having the same access to the result, I would also encourage to have if the question came from the audience, I can drop my email and contact them with a concerned IT and application team, in which they will give a full comprehensive information about IT, as those are the expert of having this. But I can just give a quick brief about it. It's like enable everyone to see the result in a very quick format, how it's coming. The IT people, they will fully give a full idea. And I'm really this question is really very important, and this will be very distinguished point even for ROCHE. If I mean the questions came from the audience, so they can drop my email. I put it on the chat box and connect them to the concerned IT department.
Okay. The last two questions, if you allow me to. One of the questions for Dr. Tariq Awed about the using for the point of care testing for at the OB/GYN department. So we can use this devices and point of care for coagulation for OB/GYN patient.
Okay, tamam. I think maybe they are referring to TEG, Thromboelestography, mainly because the other tests like the INR or the BTT, or they are not usually helpful for detecting bleeding. Thromboelestography is very nice and very helpful test, but again, the test is very complex. It's not only to run the test, it's also how to make an interpretation of the test. And unfortunately, until now, there is no software to read and analyze and give you the final diagnosis. And most of the times, it will be operator dependent. So if the OB/GYN, the surgery, the cardiac, they want to use it, it is possible to use it. It's mainly used with cardiac and liver because the bleeding tendency is more, and it was introduced long time, so they have the experience with TEG. Other services, including the hematology clinic or OB/GYN, they may use it, but my advice is before introducing or thinking about TEG, they need to know the limitation, and they need to know how to make the interpretation of the TEG system using a different cartilage or different test, because TEG is not one test, it's at least 4 tests, and the algorithm of those tests need to be collectively gathered, and then a decision made to define the bleeding and what replacement fluid, blood products that you need to do. I hope that answers the question of the operation of TEG.
Okay, it's clear. Thank you, Dr. Tariq. Regarding the INR level, the above 4. So, do you recommend to send it to the main lab to get more accurate result? And is this considered as an limitation? Should it be take care about it when you're interpreting the result from the point of care for INR?
Okay, I will ask the question from the technical point
Dr.
of view,
Malak?
and Dr. Prof. Tariq can also answer it from the clinical point of view. Most studies shows that
Yes,
there is a big discrepancy between the POCT and the lab laboratory method with the results above 4. So it's subject to confirmatory results sending to the lab, yes, until the manufacturer work more for the sensitivity of the device.
Yeah,
Prof. Tariq.
I totally agree. I think we have to understand the limitation of point of care. Point of care is a nice test, but if you don't understand the limitation, you will be put in, you will put the whole management team, including the patient, at risk. So it's been defined clearly that any patient who have an INR 4 or above, you should ignore the point of care and send a sample for the central lab to confirm. In many situations, we found that it is matching, but in another situation, we find that there is discrepancy, and the point of care had either over or underestimated the INR. But the most important, and that's what I asked usually the people for point of care, if you can keep the patients between 2 and 3 in most of the time, or 3.5 in majority of patients, I wouldn't care about the outliers of like 5 or 10% that we need to send them, because we managed 80 to 90% of the patient without a central lab. We cannot have a 100% of the patient on point of care. That's not practical, not acceptable, not safe for the patient. So keep the point of care to test for the majority, the 80 or 85%, but there will be continue to have patients with special circumstances that we will need the center.
Okay, so thank you all at the end for this interesting seminar. I would like to ask to thank the speaker, Prof. Tariq Aweda, Dr. Malak Al-Mishali, and Mr. Rani Bsiso for this interesting event, and also I would like the audience and the audience to ask the question, and also the make a good comment for the speaker about the good presentation and comprehensive information they got it. And just to remind all that the message of the lab to provide the right information to the right person, or and at the right time to make a right decision on the patient management and patient care. Thank you for Saudi Society Clinical Chemistry and SPOCT, the Saudi Point of Care Testing Association, and ROCHE for their cooperation for to make this event. And now I will conclude this this event, and thank you all, and have a good night.
Thank you all. It was a pleasure. مع السلامة.
Thank you.
Thank you.